Okay, so this is chapter 7. We're going to continue our discussion of disaccharides and then go into polysaccharides. So up to this point, we've talked about sugars in a Fisher linear conformation as well as the Hayworth, which is a circularized conformation of the sugar. And that's just a visual representation of the connectivities of the different carbons and hydroxyl groups that are connected to the monosaccharide of a sugar. However, sugars themselves actually exist primarily in the six-membered chair conformations, although we're not going to spend a lot of time on the chair conformation. So now we're going to start going over a few select disaccharides that I've chosen to cover because I think they are biologically and functionally interesting for you guys. The first is lactose, which I'm sure most of you have heard of because this is the sugar that's found in milk. It is galactose monomers and glucose monomers that are linked by a beta 1,4 bond. So it's the first carbon of galactose attached to the fourth carbon of glucose. Again, this is the sugar that's primarily found in milk. And as many of you have heard of, there's something called lactose intolerance, and this is where you're deficient of an enzyme called lactase, which breaks down lactose into its individual monosaccharides. Lactose intolerance is actually distinct from milk protein sensitivity, so some people they're sensitive to the actual proteins in milk and not actually allergic to lactose themselves. For those people that have lactose intolerance is to treat the milk with lactase, and so that's going to supplement that deficiency of the enzyme by breaking the lactose into the galactose and glucose monomers. So something that you may also find interesting is that yogurt is essentially milk that is low in lactose. So it's treated with Lactobacillus acidophilus, which yields acidophilus milk or yogurt. Okay, so another disaccharide that you might be interested in is sucrose. So sucrose is glucose and fructose that's connected by an alpha 1,2 bond. So we have carbon number 1 that is attached to carbon number 2 of fructose. And so you have both anomeric carbons in an O-glycosidic bond. Sucrose is essentially common table sugar, and it's extracted from sugarcane and sugar beets. Once it's consumed, it's hydrolyzed into glucose and fructose, where then it's metabolized and utilized for energy. One thing you might have heard of is something called high fructose corn syrup. It's actually not a disaccharide, and it's actually produced from cornstarch. So it's glucose from cornstarch, and it's partially converted by fructose by a treatment of a different assortment of enzymes. And so the reason why high fructose corn syrup is used, or is often used in the food and beverage industry, is that it contains fructose, and fructose is quite a bit sweeter than glucose, which is going to make food extra sweet. Now, there's a lot of controversy of whether high fructose corn syrup contributes to different metabolic disorders or whether it might lead to obesity or diabetes. At this point, it's still really unknown. There's a lot of conflicting research out there, but probably what is most likely the case is it's just extra sugar. So you have extra sugar, extra calories, and that is going to that can lead to obesity if there's too much sugar in the diet. Okay, so now I'm going to move on to polysaccharides. And polysaccharides are where you have many monosaccharides that are linked together. So the polysaccharides that occur in organisms usually are composed of very few types of monosaccharide components. And usually glucose is the most common monomer. So once that monosaccharide is bonded And in a polysaccharide, we call that sugar unit a residue, similar to how we call the amino acids in a protein what residue once the amino acids are actually connected together in a protein. There's homopolysaccharides. These consist of one type of monosaccharides, and again, normally it's glucose. And then it's heteropolysaccharides, and that's a polymer that consists of more than one. So it's usually glucose and some other monosaccharides such as galactose. Polysaccharides can also be branched or unbranched. If a polysaccharide has alpha glycosidic linkages that connects the different monomers together, then this will often create a branch type of structure. And that structure is going to dictate its function in that it's usually going to provide access to sugars as fuel for an organism. There's polysaccharides that are connected together by beta glycosidic linkages. And so this is going to often create a linear structure, and that linear structure is going to provide strength for an organism. So what we're going to see modeled by these examples are that the physical characteristics of a polysaccharide are determined by the type of monomers by which they're composed, so whether it's got glucose or galactose or so on, and also the type of linkages between those monomers, so whether they're alpha or beta, as well as any additional modifications that the different monomers or the different sugar residues in the polysaccharide have. And all of these different factors, all of these different structural elements are going to dictate the function of the polysaccharide in that organism. So this is just a common theme that we've seen, is that structure always dictates function in biochemistry, and that's especially true for polysaccharides. So we're going to go over some examples of polysaccharides that are involved in energy storage. Polysaccharides that are rigid polymers, as well as those that are gels. And again, the take-home point is to be able to connect how the structure of that polysaccharide is going to allow it to do its function, whether it's an energy storage, polymer, or gel. So first we're going to go over starch. So starch is the energy reserve found in plants, and it's a homopolysaccharide of D-glucose that's linked by alpha glycosidic bonds. And again, one thing about alpha glycosidic bonds in a polysaccharide is that it's very characteristic of an energy source. If something is an energy source, it probably has alpha glycosidic bonds that's attaching the different monomers together in a polysaccharide. There's actually two different forms of starch that have different degrees in branching. So there's amylose. So this is a linear type of starch that's connected by alpha 1,4 glycosidic bonds. And then there's amylopectin, in which you have alpha 1,4 glycosidic bonds that are connecting the linear chains. There's also branches that off the main sugar through alpha 1,6 glycosidic linkages, and those occur about every 12 to 25 sugar residues. The purpose of starch is that it's involved in glucose storage. So it's going to be important for storing that glucose in an organism as an energy reserve. Starch can form a helix, just like how proteins can form a helix. So you have approximately 6 residues per turn when the chain spirals. And this helix structure actually prevents extensive interchain bonding. And so it can actually bond with water and become very hydrated. Or what's interesting about the helix is that it actually forms a cage. So this cage is where iodine can bind. So if any of you guys have ever done a starch test where you add an iodine, and if there's any starch in a sample, it'll turn like a blue purplish color. That's what's happening, is the iodine binds to the cage of the helix residues. Another type of polysaccharide is glycogen. This is the energy reserve that's that we use between meals. And this is the glucose storage polymer in animals. So in plants, there's starch. In animals, there's glycogen. Very similar to starch, it's actually composed of glucose connected by alpha-1,4 glycosidic linkages. And then it's very branched. So you also have alpha-1,6 linkages at the branch points that's connected about every 10 residues in the glycogen polymer. So this highly branched structure of glycogen is really important because it's going to allow it to function by providing very fast access to the glucose residues. One thing about glycogen is that these glucose units, they can only be released at the non-reducing ends, that is at the terminal ends of a polysaccharide chain. So this is just a model of what glycogen looks like, and so only these little red dots that represent the non-reducing ends, those are the only glucose that can be released from glycogen and metabolized. And so the branching actually creates many non-reducing ends to release the glucose units. And the enzyme that's important for releasing those glucose units is glycogen phosphorylase. So it's going to cleave off single glucose residues at the non-reducing ends. And then debranching enzymes are used at the branch points. And these are just different pictures of what glycogen and starch actually look like in plants as well as the human body. They're actually stored in these dense granules. That glucose from the starch and glycogen is released when energy is needed. Okay, so cellulose is an important and the major structural component of plants, especially of wood and plant fibers. Cellulose is similar to glycogen and starch in that it's composed of glucose. So it's a homopolysaccharide chain of glucose that's linked by 1,4 glycosidic bonds. However, what makes cellulose different from glycogen and starch is that it's a linear homopolysaccharide chain, and those glycosidic linkages are in the beta conformation. So remember that starch and glycogen, those were alpha linkages. However, for cellulose, it's beta 1,4 linkages. And so you have these linear homopolysaccharide chains, and that's really important because you can have hydrogen bonding between the chains, and that's going to provide strength for the cellulose structure as well as exclude water. So that way cellulose is insoluble. And then these sheets result in a very flat sheet-like structure. Cellulose is present in plants, and the enzymes that hydrolyze cellulose, these are called cellulases. So they actually cleave the beta-glycosidic bonds. So animals, like us, we lack cellulases. So we can't actually go around eating hay because we don't have the enzymes to break down cellulose. However, celluloses are found in certain bacteria, especially including those that are in the digestive tracts of termites, as well as those of grazing animals such as cattle and horses. And that's what allows them to be able to digest grass and hay by cattle and horses, or wood by termites. Another type of homopolysaccharide is chitin. It's N-acetylglucosamine, so you have an acetyl group attached to the glucose residue that's then linked by these beta 1,4 glycosidic linkages. That N-acetylation modification is very important in the actual structure of chitin. So it allows for additional hydrogen bonding to occur between different chitin strands, and that's going to really strengthen the structure of chitin. So again, this is an example where structure dictates function. And chitin is very strong. It is used as exoskeletons in insects, lobster, shrimps, as well as the cell walls of algae, fungi, and yeast. Okay, so moving on to a different type of polysaccharide are glycosaminoglycans. So glycosaminoglycans, these are used as lubrication or gel in connective tissue. It's very viscous and it's extensively ionized. So it's modified with carboxyl groups as well as sulfonyl groups, and it then dictates the structure of these types of polysaccharides by attracting water. An example of glycosaminoglycans is chondroitin sulfate. This is the type of polysaccharide that's found at connective tissues, which provides the strength and flexibility and resistance to compression that you see in connective tissue. Just to summarize the polysaccharide examples that we've seen, there are those that are involved in energy storage. There's starch and glycogen. These tend to be connected by alpha linkages. They're branched. They have a spiral or a helical type structure, and they are highly hydrated. There are rigid polymers such as cellulose and chitin. These are homopolysaccharides that are connected by beta linkages. They're linear in structure. They tend to be insoluble and indigestible. And then there's gels, like the glycosaminoglycans. These can be alpha or beta linkages. They tend to have a highly negative charge, and they tend to be extended and unstructured as well as very hydrated. Okay, so now we're going to go into other types of complex polysaccharides and sugars. What we'll see is that polysaccharides can be further modified, and that is going to further diversify their structure and function. So glycosylation is the addition of a carbohydrate to a protein or lipids. Glycolipid is a glycosylated lipid in which a carbohydrate is attached to a lipid. And a glycoprotein is a glycosylated protein in which the carbohydrate is attached to the protein. And so there's many purposes of glycosylation in that it can help stabilize a protein. It can help the protein fold correctly. It's also involved in cellular recognition. So we're going to go over different forms of glycosylation and different types of glycolipids and glycoproteins and what those functions are. So for glycoproteins, this is when you have a monosaccharide or polysaccharide that's attached to a protein. If it's an O-linked glycoprotein, that means the sugar is attached to the oxygen of a serine or threonine. And if it's N-linked glycoprotein, that sugar is attached to an asparagine. And then it's always attached via the anomeric carbon or the reducing end of that sugar to the serine or the asparagine of the protein. So a really important example of glycoproteins are antibodies and ABO blood typing. So red blood cells, they express glycoproteins on the cell surface, and the final residue on the sugar part of the glycoprotein is going to determine your blood type. So if, for instance, your blood type, if you have A blood type, then you have N-acetylgalactosamine as the final sugar. If you have B blood type, then you have galactose as the final sugar. And then if you have AB, then you have both N-acetylgalactosamine and galactose. And then if you are O, that means you have neither N-acetylgalactosamine or galactose as the final residue on the sugar. And so if you're AB, you're considered an universal acceptor, and if you're O, then you're considered a universal donor. And so the way this works is that antibodies in your body detect and bind these specific antigens, or these specific, in this case, sugars, on the glycoprotein on your red blood cell. So our body actually makes antibodies against other glycoproteins that are not expressed on our own red blood cells. And receiving a blood transfusion of the wrong blood type will result in an immune reaction. Another example of a glycolipid are lipopolysaccharides. Lipopolysaccharides are when you have 6 fatty acids that are attached to 2 glucosamine sugars. So one glucosamine residue is then going to be the attachment site for the complex polysaccharide. Lipopolysaccharides are found in the outer membrane of gram-negative bacteria, and the lipid part of the protein is called lipid A. This is also called endotoxin, and this is toxic to human and mammals. And so this is why if we get infected by bacteria, particularly gram-negative bacteria, our bodies will actually react to that endotoxin and cause an immune response that then makes us sick.